Are you struggling with histamine and you've tried mast cell stabilizers, the enzyme DAO, etc., all to no avail? Well, I'm here to tell you that if this is your philosophy, then it's not the correct way to approach your situation. If your practitioner has been struggling to come up with an answer for you while your food options diminish and your symptoms get worse, perhaps it's time to try something more intelligent, something based on the science, something that addresses the root cause. So, let's dive into the world of the immune system and explain what your philosophy should be. If you're new, be sure to subscribe to my YouTube channel and also follow me on Instagram and Facebook as The Microbiome Expert. In my consultations, most people already know if they are high histamine. And if so, they are avoiding many of the items you see here. If you don't already know if you're high histamine, as a part of my intake, I ask about a number of these, such as alcohol, avocado, spinach, tomato, citrus, strawberries, and leftover meats. Also note here fermented foods, such as the L. reuteri yogurt, which is basically histamine in a bowl, as evidenced by some of my YouTube comments like this one, where it caused phlegm and wheezing, or this one, where it caused anxiety and agitation, or here, where it caused anxiety and sleep issues. Maybe these viewers may find this paper interesting. Brain mast cells link the immune system to anxiety-like behavior. To continue with histamine symptoms, the ones I most commonly hear about are related to the sinuses, diarrhea, skin reactions, racing heart, and mouth and throat. Maybe these sound familiar to you. So one would think that the logical solution would be to avoid high histamine foods and take some sort of medication or supplement to prevent histamine release or manage histamine after its release. And that's what most everyone does. However, you're not addressing the root cause, and odds are you will continue to go downhill over time as your food choices become fewer and fewer. So, what should we do? So let's first better understand what's going on. We'll just discuss this small corner of the very complex immune system. A number of cells produce histamine, but the predominant player is the mast cell. And mast cells not only produce histamine, and this is important, but a long list of mediators, which you can see here in Table 2. These have to do with our immune defense. Some mediators are direct antimicrobials. Others sound the alarm and recruit other components of the immune system. Others with additional purposes. The bottom line, they drive inflammation either in the short term or the long term. Obviously, the immune system is crucial to our survival. So what we want is a short, efficient, and robust immune response to an enemy, followed by resolution of the inflammatory response. We don't want chronic low-grade inflammation. If you're high histamine, that's likely what you're suffering from, and now have a hypervigilant immune system that's always looking for a fight. That's always on alert. So what's driving this chronic heightened inflammation? As we can see here, highlighted in yellow, the authors of this paper state, quote, "Mast cells are crucial for containing bacterial infection and preventing dissemination in other tissues." And they also state, quote, "The strongest functional evidence for the necessity of mast cell function to survival comes from studies involving bacterial challenges," which makes perfect sense since mast cells are mostly located in tissues which have the most exposure to the outside environment, at places of potential entry of pathogens such as the skin, the respiratory mucosa, and the GI tract. Not only are they part of our first-line defense against pathogens, but via immune signaling, they can be recruited and directed to where they are needed from their birthplace, the bone marrow. This increased presence of the number of mast cells in, say, the GI tract from immune alert signals not only increases their number, but also their contents available for release and their activity, such as increased ability to produce histamine and increased histamine receptors. This recruitment, homing, and hyperactivity concept is important to understand. And as the authors discuss here, the chronic nature of this can exacerbate the inflammatory process, as they highlight in Table 1. We'll touch on several conditions, and the innate immune function of the mast cell naturally has mechanisms to recognize pathogen load. As I've discussed before, pathogens display conserved molecular structures called pathogen-associated molecular patterns, PAMPs, which are recognized by pattern recognition receptors, PRRs, such as the toll-like receptors, TLRs, such as toll-like receptor 4, which I've mentioned in a number of videos. These toll-like receptors are on the surface of mast cells, and in humans there are 10 different toll-like receptors. Different TLRs recognize patterns from parts of various pathogens. The most researched PAMP is LPS, lipopolysaccharide, which I mention all the time. LPS is a part of the surface of gram-negative bacteria. Now, not all bad bacteria are gram-negative, but a good number are. And our immune system responds rather strongly to LPS, which is recognized by TLR4, which, as we stated, is on the surface of mast cells, among others. So when TLR4 senses LPS, it kicks off an inflammatory response within the mast cell, which could result in degranulation and release of histamine, which gives you those unpleasant symptoms. And that histamine release, as shown here, can upregulate TLR expression on the mast cell. In other words, it's a loop of signals from hell. The more bad bacteria, the more histamine release. Then, the more TLRs on the surface of the mast cell expressed at a nuclear level to respond even more so in the future to equal stimulation. And we're just talking histamine and TLR4 here, which are but two components of a slew of signals and components of the immune system. Again, there are dozens of mediators released by mast cells, which communicate with a myriad of other immune molecules. And there are 9 other toll-like receptors which interact with PAMPs from bacteria, viruses, and yeast. So to focus just on histamine metabolism is to ignore so much more at your expense. But many practitioners and patients alike, in pursuit of their own health solutions after years of frustration, focus on histamine. Many will use DAO, diamine oxidase, which is the enzyme responsible for extracellular histamine degradation. And yes, some drugs and alcohol can limit DAO activity, but in my experience, those suffering from high histamine don't drink alcohol because they react to it so terribly. They're also unlikely to take a drug which negatively affects them, and are consuming a diet of low histamine foods, all the while still on mast cell stabilizers, etc. And although a small number of people do have a genetic factor at play, this in no way explains the sheer numbers as a percentage, nor the growth curve over the years. Our genes are not changing. Other attempts to address histamine may include taking the cofactors which support DAO, based on the assumption that one may have a nutritional deficiency in one or more. Although possible, this too may have already been tried. And some may even take methyl donors, such as the overly popular 5-MTHF or SAMe, in an attempt to support the methylation pathway shown here. Of note is intracellular. Now, I'm not completely discounting these measures. I've recommended a couple myself at times when I felt it was warranted. But if they are not working in the long term, then you're going nowhere or even regressing. I have lots of success with those who are high histamine. I'm not changing genes. I very seldom recommend copper, only in those who have taken too much zinc for too long. When I recommend DAO, it's not meant to be long-term, but help with the histamine symptoms until I can do what I do, which is address the root cause, your gut microbiome. The gut microbiome is your largest source of a bacterial load. It can be dominated by good guys or dominated by bad ones, like we see here from this paper. The histamine-intolerant subjects had significantly more opportunistic pathogens in their gut microbiomes, names I often mention, such as Staphylococcus, Epicalus, Clostridium perfringens, and that family of bacteria I always say is the last family you want a high abundance of in your gut, Enterobacteriaceae. Whereas the healthy controls had significantly more bacteria from the health-promoting genus Ruminococcus and the superstar of the microbiome, F. prausnitzii. See this video for more on F. prausnitzii. What your aim should be is to have a gut microbiome which promotes health, not one which drives inflammation due to the immune system stuck on alert mode because it's constantly plugging into alarm signals which signify an enemy invasion. This is a basic survival response. 10,000 years ago, when our ancestors were drinking from a stream, eating uncooked food, suffering from a skin wound, breathing in musty cave air, or anything else, they, along with any other animal on Earth, needs to have an immune system which monitors points of entry, responds robustly and efficiently when needed, and then resolves the inflammatory state. While addressing your gut health, I aim to increase the pH in antibiotic-sensitive butyrate-producing health promoters and put in check bacteria which contribute to inflammation. For example, in this study, the researchers identified 117 species with histamine-secreting potential. None of these I aim to increase. The good bacteria I always mention are not on this list, but a number of bad guys are, such as those from the bad family Enterobacteriaceae, and as highlighted, species from the family Lactobacillusaceae, which is where your so-called probiotic Lactobacillus resides. And as these authors state, the common microbes that produce biogenic amines through decarboxylation of amino acids include Lactobacillus, which is dominant in the dysbiotic gut, by the way, Leuconostoc, Lactococcus, Enterobacter, known opportunistic pathogens, Escherichia, that's where E. coli comes from, Enterococcus, highly pathogenic bad guys, Pediococcus, Pseudomonas, known opportunistic pathogens, Streptococcus, well-known genus of many opportunistic pathogens like strep throat, Staphylococcus, known opportunistic pathogens like staph infection, Shigella, again, opportunistic pathogen, Salmonella, as in Salmonella, and Bacillus species. Lactobacillus species are known to be mainly responsible for histamine, tyramine, as in food poisoning, and putrescine, as in putrid rotting flesh, production in foods, while Enterobacteriaceae and Enterococcus species are known to contribute to production of putrescine, as discussed, cadaverine, as in cadaver, same idea as putrescine, and tyramine in foods. Not listed here is K. pneumoniae, which is a prolific histamine producer. These are the opportunistic pathogens I talk about in all of my videos. These bad actors dominate the dysbiotic microbiome. So not only are they driving inflammation, they are producing histamine and other biogenic amines. None of the good bacteria I promote were listed here. And none of the good guys I always highlight are listed here in this paper either, which looks at biogenic amine producers and degraders. Just the opportunistic pathogens I always mention, to include Lactobacillus, which is why I don't recommend fermented foods for those who are dysbiotic, and especially for those who have SIBO and or who are high histamine. A certain L. reuteri yogurt promoter writes, quote, "The issue of histamine intolerance comes front and center with our L. reuteri yogurt, since this species produces histamine, as do many other Lactobacillus species." And that, quote, "The cause is the microbial failure to metabolize histamine due to loss of histamine metabolizing species." But this logic is failed. The histamine producers and degraders share the same environment. The two tables here list many bad bacteria able to produce histamine, and even a few strains of known pathogens, such as C. perfringens, E. coli, and K. pneumoniae, were able to degrade histamine. Their logic makes no sense. The goal should be to reduce the pathogenic load from the dysbiotic gut that is signaling alarm, alarm by various mechanisms, to include LPS binding to toll-like receptor 4, which then results in more mast cells at the offended location, a higher ability to recognize invaders, and a more robust release of the immune and inflammatory mediators meant to resolve the invasion, which includes histamine. And a focus on promoting the good guys who have been sidelined in the dysbiotic gut from excessive antibiotic use, among other causes, by feeding them properly blended and dosed prebiotics so they can take control of the gut environment and do the many good things they do, one of which is to produce butyrate. And as we see here in this paper, this amazing short-chain fatty acid was able to inhibit mast cell activation by driving change in genetic expression. I hope you're enjoying the video so far. Don't forget to like, subscribe, and recommend to friends and family. Also, if you're feeling extra generous, hit the super thanks below. And the same results with the same mechanism of action are shown here, which are also shown in other studies. Butyrate is a huge friend to your gut on so many levels. This is but one. I mention butyrate in almost every video. Beyond this, the good bacteria do more than just make butyrate. They have other health-promoting benefits. And time again in my videos, I highlight how those with a given condition have significantly less health-promoting butyrate producers, while at the same time they have significantly more opportunistic pathogens driving inflammation. One such example is IBS, irritable bowel syndrome, where mast cells have been extensively studied. As these authors state, quote, mast cell alterations are prominent in IBS, including changes in mast cell number, mediator release during stimulation, and proximity to nerve tissue. And when it comes to nerve stimulation, they state, quote, the proximity of mast cells to nerve endings is closer, within 5 to 10 microns, in IBS patients versus healthy controls, which correlates with abdominal pain severity. This chart comes from my video on IBS. It shows the average microbial fingerprint for the IBS gut microbiome from my extensive meta-analysis of all human studies comparing healthy controls to those with IBS. We see in green those health-promoting butyrate producers I'm always talking about, significantly higher in healthy controls, best exemplified by F. prausnitzii, the poster child of good bacteria. Where in every IBS study I could find, F. prausnitzii was always significantly higher in the healthy controls when a significant difference was found, whereas the taxa in orange were significantly more abundant in IBS, and a number of those in orange, E. coli, Lactobacillus, and Streptococcus, we saw earlier in regards to histamine production. When we look at this meta-analysis of mast cells in the small intestine of those with IBS as compared to healthy controls, the authors quote, the number of mast cells was significantly higher in the ileum of patients with IBS. Again, number of mast cells is but one measure of their effect. And in this meta-analysis of 22 studies in 706 IBS patients and 401 controls, which looked at mast cell abundance in the large intestine, the authors state, quote, mast cells were increased in the rectosigmoid and descending colon of IBS patients. Increased mast cells were observed in both constipation and diarrhea predominant IBS. And when it comes to inflammatory bowel disease, IBD, here are a few of many papers looking into histamine's role. Findings include higher urinary histamine metabolites in IBD versus controls, ileum specimens from Crohn's patients with active disease released significantly greater amounts of histamine than specimens taken from patients with inactive disease or from control individuals, and a striking accumulation of mast cells in surgically resected intestinal strictures in patients with Crohn's. And here is my chart for the average microbial fingerprint for Crohn's, which comes from my videos on the topic. Again, we see the same pro-inflammatory bacteria significantly higher in Crohn's as compared to healthy controls. Names like Enterococcus, E. coli, Lactobacillus, and Streptococcus. Names we saw which were a part of the histamine profile. And on the flip side, we see significantly reduced levels of those butyrate producers, like F. prausnitzii and Ruminococcus, previously mentioned among others. And here is the other half of IBD, also of colitis, and we see the same story playing out. So, is it a question of not having enough histamine degraders? Or is it a question of not having enough butyrate-producing health promoters to keep the pro-inflammatory bad guys in check, so more mast cells aren't recruited and their activity upregulated to combat what is a chronic exposure to opportunistic pathogens, who, they themselves, by the way, also produce histamine above and beyond what the mast cells are doing? I want to mention autoimmune disease briefly. There is a well-researched connection between mast cells and autoimmunity. Whether it's multiple sclerosis, rheumatoid arthritis, type 1 diabetes, or another. Again, there are many facets of the immune system. Another I often mention is that of the balance between T regulatory cells and Th17. You want a tolerant immune system which is not going to react to every food protein, PAMP, or anything else in a chronic, hyperresponsive manner. The T regulatory cell tolerant immune response. The Th17 version is the one always looking for a fight. In this paper, and in a number of others, it is explained how T regulatory cells can inhibit mast cell degranulation, a good thing. But on the other hand, how histamine can reduce T cell activity and number and shift the immune system to be Th17 dominant, a bad thing. I talk about this concept in detail in my videos on autoimmune disease. And within those videos, I present this chart, which is the average microbial fingerprint for all autoimmune diseases from my large meta-analysis. You can view the names of the taxa, which are again best exemplified by the two poster children of good and bad bacteria, F. prausnitzii in green, significantly higher 100% of the time in healthy controls, which is to say 100% of the time significantly lower in those with an autoimmune disease when a significant difference was found within a trial. And on the flip side, E. coli, below the orange arrow. Mast cells are known to play a role in autism as well. In Table 1, you can clearly see in these 41 autism patients, in the last column, a long list of mast cell issues. And the authors state, quote, "We report that the apparent prevalence of autism in patients with mastocytosis, a rare disease occurring in 1 in 4,000 children and characterized by an increased number of hypersensitive mast cells in many organs, is about 1 in 10, or 10 times higher than the general population." When it comes to mast cells in the nervous system, there is an enormous connection here as well. Perhaps you've noticed that when your belly doesn't feel well, you're not 100% emotionally also. And the reverse can be true. If you become stressed, your belly can be affected. That's what these authors are highlighting here in the figure, touching on the strong interconnectivity between mast cells and neurons, or your gut and your brain. This one statement of theirs I find quite remarkable. Quote, "An estimated 70% of intestinal mucosal mast cells are in direct contact with nerves, and another 20% are within 2 micrometers. That's 2 millionths of a meter. And when a stressor degranulates the mast cells, and they release all of those pro-inflammatory compounds, not only does it impact gut permeability, but it also affects the permeability of the blood-brain barrier." These authors state, quote, "Nevertheless, they are potent cells, and even a few mast cells can release a sufficient quantity of inflammatory mediators that can affect the blood-brain barrier integrity and activate glia and neurons in the CNS. Most of us know that the brain and gut are connected, but don't understand how. There are several ways, but we're going to highlight one which is really not talked about: the permeability connection. And this is important for anxiety, depression, Parkinson's, dementia, and autism. When your gut microbiome is dysbiotic from, say, excessive antibiotic use, the bad guys are in charge. Among the many things they do is to drive gut permeability. That means whole or parts of bacteria can leave the lumen of the gut and do many things to include enter the brain. Not only does the zonulin they increase cause gut permeability, but also brain permeability. And the mast cells they stimulate also drive permeability in both. One quote from this paper is, "One of the main effects of mast cell activation is the permeabilization of the blood-brain barrier. This is done by histamine, neuropeptides, tryptase, and a number of other mediators released by mast cells. And not only does this whole process activate the microglia and astrocytes in the brain, which are so important in conditions of brain inflammation, but as the authors state, quote, mast cells express MMP2 and MMP9, which can degrade several components of the extracellular matrix, leading to rupture of the tight junctions and ultimately causing leakage of the blood-brain barrier. Altogether, this effect leads to an augmented migration of immune cells and other immunoactive compounds to the otherwise immune-privileged brain. And the same proteins responsible for preventing gut permeability, like occludin and various junctional adhesion molecules, zonular occludins, claudins, cadherins, and catenins, are responsible for maintaining the blood-brain barrier. You can learn much more about permeability from this video. The blood-brain barrier exists for a reason: to only allow in certain crucial molecules and keep out problematic ones. The same reason the gut barrier exists. We don't want the immune system which surrounds our gut or the immune system in our brain to be chronically activated by pro-inflammatory compounds which should not be there. So let's consider the short term for a moment and its connection to gut permeability and mast cells. Here we have some research on the stress of public speaking. As you can see in Figure 2, an NSAID, which is known to cause permeability, and public speech both significantly increased gut permeability in these subjects. Salivary cortisol was only increased after public speech, and the effect of public speech on gut permeability was only present in subjects with a significant elevation of cortisol. In other words, those most stressed out. In this paper, gut permeability was mediated by the immune system triggered by the stress response, except for NSAIDs. And this was proven in this paper with the use of a mast cell stabilizer. You can learn much more about the effects of emotional stress on the microbiome in this video. Our previous paper was an example of short-term stress, but what we're really concerned about is long-term stress. In this study, the researchers looked at the correlation between leaky gut, as measured by zonulin, correlated to CARS, which is a widely used tool to measure behavior in autism. As we would expect, the more zonulin, i.e. gut permeability, the more severe the behavior. And if you read the quote, these authors also talk about how the barrier that protects the brain has the same features as the barrier that maintains gut integrity, and that excess zonulin from the gut, which is generated by gut inflammation, can make its way to the brain and have the same effect there, increasing brain permeability, allowing pro-inflammatory molecules to enter the brain. In this study, the researchers looked at colonic biopsies from 32 patients with irritable bowel syndrome and 15 healthy controls, and measured blood levels of something called VIP, vasoactive intestinal polypeptide, which is one of those pro-inflammatory mediators released by mast cells and has been implicated in gut permeability in a number of studies. So let's analyze this text box line by line. Line 1, quote, plasma samples from patients with IBS had higher levels of VIP than plasma samples from controls, signifying more mast cell degranulation in response to a dysbiotic gut. Line 2, biopsies from patients with IBS had higher levels of tryptase, another mast cell mediator, larger numbers of mast cells. Remember back to that mast cell recruitment at the point of microbial invasion, and a higher percentage of mast cells that express VPAC1 than biopsies from controls. Again, that's the increased activity and ability to release contents we talked about earlier. Line number 3, and biopsies from patients with IBS, addition of Salmonella, a known pathogen, significantly reduced levels of occludin, and subsequent addition of ketotifen, a histamine-1 receptor blocker, significantly reversed this effect, showing that the gut permeability was increased by an opportunistic pathogen via histamine release. Those other bad bugs I mentioned, E. coli, Enterococcus, Streptococcus, and more, can do the same thing. This is not exclusive to Salmonella. And you're not going to win this war with histamine blockers and mast cell stabilizers. You have to get to the root cause, the bad bacteria driving this process, so they don't degranulate mast cells, which release a torrent of pro-inflammatory mediators, to include many of the ones we've seen thus far, like VIP, and many more from this paper. Table 2 highlights the powerful connection between mast cells and gut permeability, i.e. leaky gut. On the left, we see many of the mediators produced by mast cells, to include histamine. To their right, we see a number of studies connecting those mediators to gut permeability via various mechanisms. Again, we need to address the root cause of the gut inflammation, which, if unresolved, keeps one in a constant state of immune dysregulation and gut permeability, which then turns this into a system-wide problem instead of localized to the gut. Suffering from histamine issues is no fun. Many of the people with whom I work have come to me after struggling with it for many years, with all sorts of reactions, lots of food avoidance, and failed therapies. High histamine can be a bit of a challenge to treat. Some of my people glide right through their protocol and begin reintroducing foods even sooner than I'm comfortable with. Some take longer. While going through the protocol I have built for you, weird things can happen. Some symptoms may get worse before they get better. You'll have to be a trooper, which is why I don't offer up a $25 pre-made protocol for high histamine. You'll need the extra support I offer from my consultations. It took you years to get to this point. We have to calm down your immune system, and that doesn't happen overnight. Just bear in mind, I address the root cause, not the symptoms like everyone else is focused on. So, if you're ready to try something more intelligent to address your long-term needs, then go to my website, themicrobiomeexpert.com, and schedule a consultation with me today. If you liked the video, don't forget to subscribe. Also, somewhere around here, you can go to my website where you can schedule a consultation with me. You can also view the protocols, and here you can watch the next video.